All right, today we are diving deep into one of the most incredible stories in all of science. We're talking about the molecular basis of inheritance. You know, how the instruction manual for every living thing is stored, copied, and, most importantly, actually read.
And it all really kicks off with this one huge question. I mean, just think about it for a second. How do you go from a single tiny cell to, say, a giant redwood tree or a person? All of the information, every single instruction for that amazing journey is packed inside that one microscopic cell. It tells you there has to be some kind of master blueprint, right? A code for life itself.
And of course, that blueprint is a molecule I'm sure you've all heard of. It's the absolute star of our show today, DNA.
So what is it really? Well, its full name is deoxyribonucleic acid, DNA for short, and it's what we call a long polymer. Just picture a super long chain made of repeating parts. This amazing molecule is the genetic material for almost all life on Earth. It holds every single instruction for how an organism is going to develop, how it's going to function, and how it's going to reproduce. It's the whole shebang.
Now get ready for a mind-bending fact about scale. If you could take the DNA from just one of your cells, just one, and stretch it all the way out, it would be about 2.2 meters long. Yeah, that's taller than most people. All of that is crammed into a space you can't even see without a microscope.
Which, immediately leads to the next logical question. How in the world do you fit over 2 meters of this stuff into a nucleus that's ridiculously tiny? Well, the answer is just brilliant, super efficient packaging.
The solution is just a masterpiece of natural design. You see, the DNA molecule has a negative charge, so it wraps itself tightly around these little proteins called histones, which have a positive charge. Opposites attract. And this creates a structure that, when you look at it, looks exactly like beads on a string. It's an absolutely elegant and unbelievably compact way to store all that information.
Okay, so we know how it's packed in there. But what about the structure of the DNA molecule itself? That amazing packaging is only possible because of its famous, iconic shape, the double helix.
Ah, the double helix. You've definitely seen this shape before. Back in 1953, James Watson and Francis Crick put together this elegant model, using absolutely critical X-ray data from researchers Maurice Wilkins and Rosalind Franklin. And let me tell you, their discovery changed biology forever. So the beauty of this double helix is that it follows some really simple but very strict rules. The easiest way to think about it is like a twisted ladder. The two long sides of the ladder are the backbone. And the rungs? Well, the rungs are made of pairs of molecules we call bases, A, T, C, and G. And here is the golden rule. A always pairs with T, and C always pairs with G. This perfect pairing is what gives DNA its uniform shape, and as we're about to see, it's the secret sauce for how it copies itself so perfectly. And this is where it gets really clever. The true genius of the double helix isn't just its shape, it's that the shape itself screams at you exactly how it can be copied. And Watson and Crick knew it from the very beginning. This right here is one of the biggest understatements in the history of science. In their famous 1953 paper, they basically said, hey, by the way, this AT and CG pairing thing we just figured out, yeah, it kind of tells you exactly how this whole thing replicates. Incredible.
So, this copying process has a name: semi-conservative replication. It sounds a little technical, but the idea is just beautiful. The DNA ladder basically unzips right down the middle, and each of the two original strands becomes a perfect template to build a brand new partner strand.
So look, you start with one parent DNA molecule. It separates, and just like that, you have two new daughter molecules. But here's the key. Each of those new molecules is actually a hybrid. It's got one of the original strands and one brand new one. That's what semi-conservative means. Half is conserved. It's the cell's foolproof system for making a perfect, faithful copy every single time.
Okay, so we can store the blueprint, and we can copy the blueprint. Awesome. But how does the cell actually read the instructions and start building things? That is the next crucial step in our story. To get the job done, the cell uses a slightly different molecule called RNA, or ribonucleic acid. Now, if DNA is the precious master blueprint that's kept safe in the nucleus, think of RNA as the working copy. It's like a temporary, disposable photocopy of a small part of the blueprint. It's single-stranded and less stable than DNA, which actually makes it perfect for its job as a short-term messenger.
So the first step in reading the blueprint is called transcription.
This is where a little segment of the DNA is copied into a molecule of messenger RNA, or mRNA. I absolutely love this analogy. Imagine going to a library to see a rare, priceless cookbook. You wouldn't check the whole book out, right? No, you just make a photocopy of the one recipe you need. That's exactly what transcription is.
And step 2, that's translation. This is where the cell takes that photocopied recipe, the mRNA, over to its molecular kitchen, which is a little factory called a ribosome. The ribosome reads the code on the mRNA, and based on those instructions, it starts building a protein, piece by piece. The recipe is now being used to actually cook the final dish.
So the whole flow of information is wonderfully simple when you think about it. The permanent instructions in the DNA are transcribed into a temporary RNA message. That message is then translated into a functional protein. This simple little diagram, well, it represents one of the most fundamental processes in all of biology.
And this fundamental flow, this core principle, has a name. And it's a pretty grand title for a pretty grand idea. It's called the central dogma of molecular biology. Francis Crick, the same guy from the double helix, proposed it. And it simply states that genetic information flows in one direction. It goes from DNA, which can copy itself, to RNA, and from RNA to protein. It's the absolute core rulebook for how life puts its instructions into action.
So let's just put it all together one last time. Step 1 is replication. The entire DNA blueprint gets copied, usually before a cell divides. Step 2 is transcription. A small section of that blueprint gets copied into a messenger RNA. And finally, step 3 is translation. That RNA message is read to build a specific protein that goes off and does a job.
And that's basically it. This beautiful, elegant, three-part system is the engine of life. It's how information becomes reality. It's happening inside every one of your cells right this second, and it's the shared foundation for just about every living thing on this planet.
It's an unbelievably robust and amazing system, but, it's not always 100% perfect. So the last question I want to leave you with is this. If this is the rulebook, the ultimate code for building life, what happens when there's a typo? Because that one question, well, that's the key to understanding everything from genetic diseases to the entire beautiful process of evolution.